Let's get started on Chapter 1, which is an introduction to microbiology. In this chapter, we'll talk about what microorganisms are, the groups of microorganisms, how they are classified, and what the basis of that classification is. Then we'll also talk about the brief history of microbiology and some of the major themes and theories that have arisen and how that has translated to lots of different fields of science and interest. So getting started. So there are a lot of microorganisms that are present on our bodies, around in our surroundings, and they are prevalent and present everywhere. What makes a microorganism is basically the size. So microorganisms are those that are living creatures that are too small to be seen with the unaided eye, which means they are so small that we cannot see them just with our eyes. We need some instrument or device which will magnify them so that we can see them or at least visualize them. Now, there are a lot of microorganisms that are included, and these are not just prokaryotes. You also have eukaryotic microorganisms. The name microorganism, if you break it up, it is micro and organism. So the micro part tells you that this is about size, and the size, if you look at meters and think about micrometers, the microorganisms are in micrometer lengths. A micrometer is 1/1,000,000th of a meter. So if you're looking at a microorganism, you're seeing something that is 1,000,000 times smaller than a meter stick. So if you were to break up the meter stick into 1,000,000 different parts, each of them would be 1 micrometer. And lot of the bacteria that we see in our slides and pictures, these are in the 2 micrometers across and 5 micrometers across range. They are smaller microorganisms. They are much smaller microorganisms, and they can be slightly larger also. But the ones that are smaller than bacteria, they can be in the nanometer range. So a nanometer is a billionth with a B. So it's, if you take a meter stick and take it into a billion pieces, you will get 1/1000,000th or a nanometer. So you can have microorganisms in the nanometer range as well. So the nanometer microorganisms are usually fungi, the viruses. Viruses are, so if you look at a bacterium, a virus could be 1,000 times smaller than the bacterium, and that would be the nanometer range. Now, viruses are unique in that they are different from the rest of the microorganisms, but you also have viroids and prions, which kind of are derivatives of infectious material. So viroids are just infectious RNA and prions are just infectious protein. So we have microorganisms which run the gamut from bacteria, fungi, protozoa, microscopic algae, and then viruses, and then even below viruses. So we will discuss each of these groups in a little bit more detail as we go along. Right now we're just getting introduced to these few groups. The microbes in our lives, they are not all disease causing. A few of them are, and these are the ones that ail us the most. Literally, they will make you ill, and they are disease producing. So when you are encountering microorganisms, sometimes they are only obvious because we have been hurt by them, but they are not all bad, and a lot of them have this very important job of decomposing organic waste. So all of the creatures that die, they have to be decomposed back to their original elements so that they can be recycled and back into the soil so plants can utilize them, and then plants produce the food for the primary consumers, and then primary consumers are in turn food for secondary consumers, and so on. So unless microorganisms decompose all organic matter, this cycle will not continue. So they are a very important link in the food chain. They also are, some of these microorganisms are able to photosynthesize, which means they can directly take the carbon dioxide from the atmosphere, use oxygen. Sometimes they don't even need oxygen, and they can produce carbohydrates, which is what photosynthesis is. And those carbohydrates then are available to the rest of the consumers. They can also produce chemicals like ethanol, acetone, and vitamins. Now, I'm sure ethanol might look unfamiliar, but if we talk about beer fermentation, then it becomes obvious what we're talking about as far as chemical products. You also have fermented foods such as vinegar, cheese, and bread, which are generated by microorganisms. You probably eat a lot of yogurt, or you consume some probiotics when you're given antibiotics. So all of these are the beneficial microorganisms that are helpful to make products for us, also help us digest, and help us keep safe and increase our immunity. There are other interesting products that could be used in manufacturing that are based from microorganisms. So like cellulase, which is used to digest cellulose, which you find in plants. And then that can be a fiber cellulose, which is broken down, and you can make separate products. You can also make microorganisms produce such proteins such as insulin, which is obviously useful for diabetes treatment in humans. So the gamut of what microorganisms do is it runs the range, and you have pathogenic, and you have helpful microorganisms, you have really useful for products, and then manufacturing and biotechnology products. So microorganisms are going to be an important part of all of our lives, and it is important that we understand that they are not all bad. So some of the interesting things that you get from microorganisms, one of them is trichoderma, is a microbe which will produce the enzyme cellulase, and that cellulase can be used on denim fabric, and that can give it that stone-washed look, and this is then called biostoning. You can have cotton production, and you can have bleaching done with mushroom peroxidase. You can have indigo production through E. coli, and you can have a bacterial plastic generator. So as you can see, humans have figured out there are interesting enzymes and products from microorganisms that can make our products better, or unique, certainly. So this is a picture of a microorganism that is able to digest denim or cellulose. So cellulase producing, and then that gives that faded biostoning look. So why do we need to know about these microorganisms? One of the important things is that it allows humans to prevent food spoilage. So a lot of the initial history of microorganisms started with folks noticing that food left outside got spoiled, it got rotten, it was not consumable anymore. So if we knew what was causing that spoilage, maybe we can prevent it. So that is an important knowledge to have. And another very important knowledge is to be able to prevent disease. So if we know a microorganism is causing a disease, and we know something about that microorganism, maybe we can do something about stopping it or preventing disease. And then right now, it is very important to understand what is the cause and how it is transmitted, and that can allow us not just to prevent disease but also to treat and to prevent further spread of disease. So when you see these slides which have these questions which ask you to check your understanding, these are what you will take down and answer for me so that I understand that you have gone through this video. So when you look at microorganisms, the first thing you want to know is what kind of microorganism is it? Where do I put it into this whole group of microorganisms? So we mentioned there are bacteria, there are protozoa, there are worms, etc., there are algae. So where is this particular microbe that I am interested in falling into place? So this question was kind of studied on and tried to be understood well, so that there was a naming system that came into place, and Carolus Linnaeus established a system of scientific nomenclature. Now obviously at that point it was not microorganisms he was naming, but he was naming birds, flowers, things like that. But that naming system has been extended to every living thing, and so every organism has two names now: a genus and a species, or a specific epithet. So a genus and species is going to identify a microorganism or any other organism down to a few very unique classifications. So Homo sapiens, which refers to humans, is going to be uniquely telling us this is about humans. So if we talk about a plant like maybe hibiscus, Rosa sinensis, that says okay, Chinese hibiscus, that is talking about a flower. So this is going to be a unique way to quickly identify which microorganism or which organism we're talking about. So each organism has two names, and that's why this is also called the binomial nomenclature. So you will see references to scientific nomenclature, to binomial nomenclature, or to Linnaean nomenclature, and all means the same thing, that each organism has a genus and species two-word name, and that identifies it uniquely. So the genus and species is at the tail end of a five kingdoms classification system. And so if you look at the hierarchy, you have kingdom, phylum, class, order, family, genus, and species. And these are some videos which can help you understand how this system is set up. But for now, understand that there is an overarching group of classification, and so every living thing falls under that. Then there are three subheadings in which these are the domains. So there are three domains. On the basis of those domains, you will further sort living things into whether it's a bacteria, archaebacteria, or a eukaryote. So once the three domains are established, under the domains come five kingdoms, and in those five kingdoms, four of the kingdoms are eukaryotic, and one kingdom is prokaryotic. The one kingdom that is prokaryotic has the bacteria in it. The five kingdoms that are there, four of those are eukaryotic, and then you have the animals, the plants, the fungi, and the protozoa. These are the four kingdoms which fall under the eukaryotes. Now there'll be visuals later on in the video, so don't worry about it just yet. So the three domains, these are, like I mentioned, the archaebacteria, bacteria, and eukaryote. So before we wind up the nomenclature and the naming and go on to domains, there are very important rules to understand while naming any organism. And that nomenclature is that you have to write the name in italics. So if you're typing it out, it has to be italicized. If you're writing it out, you have to underline it. So if you are typing it out, you italicize the first genus, second species. The genus is always capitalized and the species starts with a lowercase letter. And these are usually Latinized or Latin names for these microorganisms because that was the language of science to start with. The names in themselves might be descriptive or they might honor a scientist who has discovered them. So we'll run through a few examples. So if you look at E. coli, and this is quite familiar to most of us, it honors a discoverer, Escherichia, and you have Escherichia coli. So E. coli, that's what you've probably seen. And the coli tells you that this was first noted in the colon or the large intestine of humans. So here it does not show that, but this should be italicized. The E is capitalized, so because this is the genus, and the C should be lowercase, and these two either need to be underlined and even when you're underlining them, this has to be a separate underline. So if you have the underline, it has to be E. coli. And it has to be italicized while typing. Staphylococcus aureus, again, this first letter is capital, this which is the genus, and then the second, which is the species, is lowercase. Staphylococcus aureus. And this describes how the bacteria look like under the microscope. The staphylococcus look like clustered round or spherical cells. So clustering is defined as staphylo arrangement, and the cocci, it tells you that it is a circular microorganism. Now when these are grown on plates, they look golden or yellowish, so that's why they appear to look gold colored, and therefore aureus. Aureus, the AU is the element symbol for gold or aurum. So Staphylococcus aureus tells you a few things. One, that it has a staphyloid arrangement, in that these cells are form grape-like clusters. It is circular, so coccus. And aureus, the growth on nutrient plates when cultured looks golden. So you can take E. coli or staphylococcus and write it in a document. The first time you write it, you have to either italicize it or have separate underlines and write the whole genus and species out. But once you just start writing more in the document and you're just referring to it, you can make it E. coli. So you can have that dot come in, and now it's referencing the fact that you've already mentioned the first name. And with Staphylococcus aureus, you can, the second time around, you still have to underline it, but you can abbreviate the first letter or the genus. So again, checking your understanding, can you distinguish a genus from a species? And if you are given a few names, you should be able to say which is which and how to write it out properly. Okay, so the next set of things that we will discuss is the types of microorganisms. So bacteria, archaebacteria, fungi, protozoa, algae, viruses, and multicellular animal parasites. All of these are microorganisms. Now with the multicellular animal parasites, you might think, oh, I'm sure I can see worms, and how do they get to be microorganisms? Because they are visual, I can see them without needing a visual aid. So it is not so much that the worm itself is the microorganism, but some phases of its life are. So maybe it's in the early stages when it's very small, and at that point it's easy to transmit also. At those types of stages, these are microscopic, and so that's why they are also classified into microorganisms. So we'll look at all of these groups independently. So this first picture is of bacteria, and this is the typical bacterial size and shape you see. This is a rod-shaped bacterium. This is showing fungal spores. So this is growth. The growth is spread out like a mesh, and then you have fruiting bodies called sporangia with spores on them. The spores themselves are seeds. So this whole thing is maybe what you would see if you looked at bread mold under a microscope. You'd see the hypha, which is this mesh-like network. You would see the sporangia and the spores. This third picture is the amoeba, and amoeba has sort of an amorphous shape. It is shapeless, but it can extend its cytoplasm and engulf something that it's eating. This is a picture of algae, and these single green dots are the algae, but when they live together, they can form a colony, and then they kind of become more visible like that. In this last picture, you're looking at a human cell which has these red viruses sitting on them. So if you look at the size range, now SEM refers to scanning electron micrograph, and LM refers to light micrograph. So the types of microscopy will help us see something in much more detail and clarity. Any electron microscope can do a much more detailed job than a light microscope. Also, with the scanning electron microscope, you see the outside contours. So if you look at the range of this microorganism, it is telling you 3 micrometer. And so these are much bigger, the fungi, so they go into the 50 micrometer range. And the protozoa, and this is showing you amoeba, is also equivalent almost to the fungal size, which is 50 micrometers. But you can see that each protozoan is going to be quite a lot bigger than a bacterial cell. So if you just take that bar and put it across here, that is 3 micrometers. And maybe it is 1 micrometer across. If you take that same bar and you look at the fungus, you can see that each fungal sporangia or the growth here is about 50 micrometers this way. So already you have 10 to 15 times larger sizes here. This protozoan may be 50, 100, 150 across, and so much bigger than even the fungi. Now we are here in the micrometer range again, but even 10 times larger than the protozoan. But now again, consider that this is not one algae, it is a group. So it is a colony. So the colony, when all of these individuals come together, can be a much more apparent structure, and visually you can see them more easily, even using the light microscope, which has much less magnifying power than an electron microscope. Now, if you look at the virus, it's showing you nanometer range. So now we are not even in that micrometer range. We're 1,000 times lower with the nanometer range. So these viruses, they could be 1,000 times smaller than bacteria. And so these are much smaller, and if you look at the cell that they are attacking, that is much bigger, and a lot of these have infected that one cell. So this is important to understand as far as size goes, that these are the range of sizes, the nanometer, the micrometer, and if you took it times 10, this would now become the meter range because you have 1,000 micrometers becoming 1 meter. 1,000 micrometers would become 1 millimeter. Okay, so looking at bacteria, and the bacteria are important. Classification is that they are prokaryotic, which means they don't have a nucleus. So pro means before and karyote means seed. So before the seed or pre-nucleus, they are the microorganisms which are much simpler and they do not have a nucleus. All bacteria are single celled, which means one cell is the whole microorganism, the entity, the whole organism is that one cell. It lives independently. It is able to do all of its metabolic activity by itself. It does not depend on any other cell for its life or nutrition. And they have this important material called peptidoglycan, which is a important component of their cell walls. The peptide part tells you that there is some level of protein in it. The glycan part tells you that it has a sugar component also. So these are unique sugars and protein combination that are found in the cell walls of bacteria. So the walls of bacteria have peptidoglycan, and that's a very unique thing that you would not see in any other microorganism or organism. So peptidoglycan cell walls are an important feature of bacteria. Also, they divide via binary fission, which means you take one cell and this splits and gives rise to two cells, and that is a mode of reproduction. This then will give rise to two more cells and so on. It'll keep making more cells. So division by binary fission leads to more microorganisms. They get nutrition from organic or inorganic sources or photosynthesis. So they have a lot of different ways of getting nutrition. They live in all kinds of environments, so they have adapted to those different kinds of environments. And we will look at the nutritive basis in a different chapter. Now, fungi. So fungi are eukaryotic, which means they do have a distinct nucleus, and along with that distinct nucleus there come organelles, and organelles help divide the labor or divide the work in a cell into different parts, and each part or each organelle or has different jobs to do. So the genetic material is housed in the nucleus. Maybe there's a mitochondria that is taking care of energy metabolic activities. So this cell has a lot more complications or complex lifestyle than a prokaryote. A prokaryote would just have the membrane, it would have the cytoplasm, it would have genetic material. The genetic material, however, is not enclosed in any membrane, and so this is kind of just in the center of the cell, and then the cytoplasmic material, the protein synthesis machinery, ribosomes, all of these are present, but there are no separate housing units for them. And that's what's different with a prokaryotic cell and a eukaryotic cell. Eukaryotic cells have that special nuclear region. They have these special organelles which do different jobs. They also have cytoplasm, which is rich in different kinds of materials. But most definitely the important part is that it is a membrane-bound nucleus. Now, the fungi have another kind of distinctive material in their cell wall, and that's a polysaccharide called chitin. And what it also has, much like starch, it's a carbohydrate, but it has a certain different kind of chemical bonding in it. Even though it is made up of glucose units, it also has certain mineral elements in it. It can absorb organic chemicals for energy. The fungus can, and that is how it gets nutrition for itself. Now, fungi, they range from being a single cell organism to a multicellular organism. So that means that this one cell is the whole organism. It can live by itself, or you can have fungi which have a lot of cells and they form part of a bigger organism, and that is the whole organism. So maybe think of this as one single unit. This is several single units, but they are part of one microorganism. So this is a unicellular yeast, and maybe you've seen mushrooms, and that would be a multicellular fungus. So molds and mushrooms, these are multicellular. You can have a lot of fibrous mycelia, so thread-like structures. These extend the fungus, and each of these units is called a hypha. So this mesh network can then give rise to fruiting bodies, and you can have lots of seeds of the fungus, or spores as they are called, in there. So if you have mold on your bread and you dust it off and this fine dust of black or green stuff comes out, those are the spores of fungi. So those are the spore holders, the sporangia, and this of course is Alice, and the mushrooms probably made her high, and she had that wonderful experience that Lewis Carroll mentioned, going through the rabbit hole and interesting places. So you've got fungi, which is a eukaryote. Another group of eukaryotes that is part of the microorganisms are these protozoa. So one of the pictures that we saw earlier was the amoeba. The amoeba is a protozoan. So these are eukaryotes, which mean again that they have a distinct nucleus that is membrane bound. Now these protozoa can either absorb or ingest organic chemicals, and that's how they would get their nutrition. So that picture showed the amoeba extending its cytoplasm out, and this can form a covering and enclose its food particle. And so these are called pseudopodia, or false feet. So these extend out. These are cytoplasmic extensions, and the cytoplasmic extensions can help it engulf food. It can also have hair-like extensions on the outside, or a long protein-based tail. All of these can help amoeba move. So they can either be free living, in which case they get their own nutrition, they are not dependent on a host, or they can be parasitic and they can derive nutrients from a living host. So this is again that picture of the amoeba, and you can see how it's spread out like that. If there's a food particle, it will extend its false feet or pseudopodia and engulf it and digest it. You have a lot of different kinds of protozoans. So amoeba is one kind. The parasitic one, the most clearest example is the malarial parasite called Plasmodium. The Plasmodium parasite is transmitted to humans via the bite of a mosquito. So part of the life cycle of that protozoan is in humans and part of it is in the mosquito. It doesn't actually infect the mosquito, but it needs to be in a warm-blooded host like humans to further multiply, and it usually does that in the red blood cells, and the red blood cells are the living host cell where the malarial parasite resides. A group of prokaryotes, which are an important part of the microorganism array, are archaebacteria. Now, archaea or archaic refers to something old, and so these are grouped into that category because they live in extreme environments. And the idea is that because the earth had extreme environments at the beginning when it was still in the formative stages, and there were a lot of either ice cold temperatures or really hot temperatures or really salty conditions. So the bacteria that could grow at that time or lived at that time were living in extreme conditions. So the bacteria that can live in those extreme conditions are therefore called archaebacteria, archaic. So maybe having adaptations would allow them to live in extreme environments. They are a lot like bacteria, but they are also different in significant ways. The similarity is that they are prokaryotes. So prokaryotes are going to have no nucleus, and they are going to have genetic material which is standalone. It sits in the middle of the cell, but they don't have peptidoglycan, that special protein sugar complex we talked about in bacteria. They have something which is similar to peptidoglycan but not the same. So there is something called murein, murein, which is peptidoglycan's other name. So prokaryotes, like archaebacteria, can either have archaebacteria will have pseudomurein, which means it looks like peptidoglycan but it isn't, and the bacteria have murein. So these, like I mentioned, they live in extreme environments. So you'll find them in really hot places, really cold places, really salty places. Maybe they are in sulfur springs. They are then called extremophiles. So methanogens, methane producers, extreme halophiles, these are they can tolerate high salt conditions. So anywhere like in the ocean or in the Dead Sea where there is a super high concentration of salt, you will find these archaebacteria. Extreme thermophiles, and as you can imagine, the thermo tells you heat and the phili or the phile tells you heat loving. So these are heat loving microorganisms, so you might find them in thermal vents or hot sulfur springs, etc. So any extreme environment, anything living there most likely is an archaebacteria. So these are archaebacteria grown on a plate. They're mighty difficult to grow on plates, but if you do grow them, you can see different colors, as you can see if they were in hot springs or high salt conditions. They give these beautiful colorations to water bodies, or this is national parks that might have sulfur springs. So coming to another eukaryotic microorganism, which is algae. Now algae also, you can run the gamut from it being a single small cell to a part of a multicellular organism. So the important thing is that these have cellulose cell walls. Cellulose is a carbohydrate made up of glucose, much like starch, but the bonds between the glucose are different, giving it a very different structure. Now, while humans can break down starch and use it for energy, we cannot digest cellulose. The cellulose, because even though it is a glucose bonded to another glucose, much like starch is, it is not bonded in the same way. We don't have enzymes that break these cellulose bonds. An enzyme that can break a cellulose bond is called a cellulase, and we don't have those. But cows in their gut have microorganisms that have cellulase producing microbes, and these can then digest the huge amount of grass that the cow eats. And so there are other bacteria also outside free living, not just inside the gut of animals, which have the ability to break down cellulose. But algae themselves, their cell walls, they are eukaryotic, which means they have a nucleus and all the organelles, and they also have a cell wall that is based off of cellulose. They can be found in freshwater, saltwater water bodies, or in soil. And they can photosynthesize, which means they can use the light energy from the sun and carbon dioxide from the air and generate carbohydrates, which they can then break down for energy. So in the process of photosynthesis, they can produce oxygen as a byproduct in addition to the carbohydrate. So these also are then part of what could be called primary producers. So primary producers, you have plants which also photosynthesize, but these are not microorganisms. However, the primary producer, which is a microorganism, is algae. So this is a colonial algae. So you can either have unicellular algae, in which case they live alone and independently, but they can also form groups, which are called colonies. They can still be independent, but they just form a group. They're not one whole organism. Or you can have algae, which are multicellular and part of an organism. So like, seaweed or kelp, those are multicellular. Now coming to a very important group of microorganisms, which are viruses. Now viruses can also be thought of as acellular, which of course that means that they don't have the same kind of cells we've talked about, the prokaryotic cell and the eukaryotic cell. They don't have that same structure. There's no such organization. They are very simple infectious particles. But the fact is that they are infectious particles, and that is what makes them living. If they did not have a living host, they could not multiply. They could be on surfaces, but they need a living host to infect and then to multiply, and they need that because they don't have that cellular structure. They don't have an ability to produce proteins, to divide, to make any of their cellular products. For that, they will go into a host, take over the host cell machinery, and then be able to direct that host to making more viral particles. So the idea is a virus is going to be living only if it finds a proper host. So once it finds the host, of course, then it is able to multiply, make more copies of the virus, and then spread to other areas and other hosts. The virus at the core has some genetic material, which could be DNA or RNA. Most eukaryotes have DNA as their genetic material, and all eukaryotes and prokaryotes actually have DNA as their genetic material. The only living things or sort of living things that have any other optional genetic material are viruses. They can either have DNA or they can have RNA as their genetic material. So first, they have a genetic material that is then enclosed in a protein shell. That protein shell may or may not have an additional covering, which is oil-based or lipid-based. So you have genetic material, and that's what's going to direct the host to do all kinds of things for the virus, but it comes packaged with a covering, a protective covering, which is called the protein coat or capsid, and that may or may not be covered by an additional covering. So what you see here is that kind of viral particle, the genetic material covered by a protein coat, covered by a lipid envelope. And this is showing you virus like the avian flu virus, and these H and N are the ones that give you the H1N1 kind of designation. These are specific carbohydrate protein spikes that are present on the outside of the virus. They are unique viral protein carbohydrate complexes, and they help attach to hosts. So once this virus, and this is a human virus. Now if it's a bacterial virus, which is called a bacteriophage, it doesn't have this envelope. Human viruses are the only ones that have these envelopes. The DNA or RNA is enclosed in a protein. So at the most basic, that's what a virus will have. And so outside of a living host, they can't do anything. But once they find a host, they will take over the host. They'll ask that host to start making more viral particles. And in that process, because the viral load of making all of these things is higher, the cell starts to feel sick, to feel loss of energy, and it might die at the end of this infection, or it might be severely debilitated. So effects of the viral infection are almost collateral damage as far as the virus is concerned. Its main job is to make more copies of the virus. So the two things that which make a virus living is its ability to infect and its ability to reproduce. So in both of these things, it is showing that it is a living thing. In other things, like it being inert outside a living host, it can be crystallized, it can be dried. All of those things make it almost non-living. But the minute it finds the right and appropriate host, it will infect and it'll make more viral particles. Now with any given virus, it has a very specific host to virus relationship. So if it is a bacterium, it'll have a specific virus that only targets that bacterium, and that's mainly because they have to match up, and this has to then allow entry of the virus. And once the virus has entered, it has to have the right keys, genetic and protein keys, to take over the host cell machinery. So all of these are very specific arrangements, and that is why the virus host relationship is very specific. So this is a picture of the coronavirus that we've been seeing, and this is HIV virus infecting its host cell. Now the Zika virus is slightly different from either the coronavirus or the HIV virus in that it has a zoonotic host, which means it has a carrier, which is another animal, which is not its final host. So it is carried on mosquitoes. The mosquitoes introduce it into the human, and that's where it multiplies. But the virus itself will not infect its vector host, which is the zoonotic host, or the the mosquito in this case. Ebola is a disease caused by a virus, but the it's hemorrhagic, which means it causes bleeding in the host, and this is way more infective and lethal, or the fatality rates are higher than a lot of other influenza type viruses. But because its body fluid exchange is not as easily done, it can still be contained with proper precautions as compared to a flu or airborne sort of virus. So this is the hemorrhagic fever, and also it is thought much like any of these new viruses that have come about, to have had a non-human origination, maybe a bat or another mammal, and then it hopped species and became more infectious in humans. Now coming to the last category of what microorganisms we've noticed or noted is the multicellular animal parasites. So you can have multicellular animals as the like helminths, which are microorganisms in early stages of their life cycle. So parasitic flatworms and roundworms, these are called helminths, and some of the early stages like the scolex stage and the stage which is cyst stage are too small to be seen with the naked eye, in which case then they are called microscopic. So this is a close-up of a tapeworm, which is found or can cause infections in humans, and it causes massive gastrointestinal disorders. This part of its life cycle where it is like this worm is not the microscopic part. It has a small head like this, which anchors into the intestine, and then it pokes holes in the intestine and causes diarrhea and loss of nutrition and causes massive dehydration. The uncooked pork is a vehicle for helminths, in this case of tapeworm. This is the roundworm, and it has two distinct forms, male and female, and the roundworm is going to be one of those again smaller stages. When it's still a cyst stage, it would be microscopic, but then it can be infectious, and fully grown it's visible. So again, to check your understanding, look at these categories and try to figure out which one of these are prokaryotic and which one of these are eukaryotic. So I will end my recording here, and we will continue from the classification of microorganisms.